Here we'll learn some basic principles of infectious disease. In part 2, we'll delve into infectious disease transmission, prevention, and treatment. To begin, start a table and to note that a pathogen is a microorganism capable of causing disease. We can broadly divide pathogens into bacteria, which are prokaryotic, viruses, which are non-cellular, and three eukaryotic types: fungi, including yeasts and molds, protozoa, and helminths. Denote that virulence is a measure of how much damage a pathogen can cause. It's determined by virulence factors, which vary by organism. For example, bacterial adhesions and toxins facilitate host invasion and pathogenic effects. Infections can be asymptomatic, also called subclinical, which means that the pathogen exists but does not cause disease symptoms. Individuals with asymptomatic infections are called carriers because they can carry and therefore transmit the infection to others. Lastly, define exogenous versus endogenous sources of infection. Exogenous infections occur when the pathogen is acquired from the external environment. For example, influenza is caused by a virus that individuals catch from viral particles in their surroundings. Endogenous infections are caused by opportunistic pathogens that are members of the host's normal microbial flora. For example, the bacteria Staphylococcus aureus, which can cause significant pathology, is commonly found in the human nares. Be aware that the distinction between exogenous and endogenous infections is sometimes difficult to discern and can depend on what's considered normal microbial flora. With these definitions in place, let's illustrate some common routes of pathogen entry. First, indicate that entry through the skin is typically blocked physically by the stratified epithelium of the epidermis, and chemically by antimicrobial defensins and fatty acids. When these barriers fail, microorganisms from the surface of the skin, such as the bacteria Staphylococcus aureus, can gain entry. Other organisms take a more aggressive approach. For example, the larvae of the parasitic flatworm Schistosoma release enzymes to destroy keratinocyte bonds, thus actively creating their own means to entry. Next, indicate that the respiratory tract is protected by mucus and cilia that clear harmful materials, and by phagocytic alveolar macrophages in the lungs. However, some microbes have adaptations to override these protections. For example, the influenza virus uses hemagglutinins to bind respiratory epithelial cells, and the bacteria Mycobacterium tuberculosis survives the phagolysosomes of the alveolar macrophages. Next, indicate that the epithelial lining of the gastrointestinal tract provides multiple sources of protection. The gastric glands produce a viscous layer of mucus, acids, and enzymes to trap and disable pathogens. Furthermore, the normal gut flora prevents pathogenic microorganisms from colonizing. However, when antibiotics kill the microbial flora, Clostridium difficile can flourish, leading to colitis and diarrhea. Poliovirus is an example of a microorganism that gains entry by hijacking normal host physiology. It appears that the gut lymphoid tissue, which naturally pulls antigens into the circulation, may likewise take up poliovirus and facilitate its spread. Lastly, indicate that the urogenital tract has multiple sources of protection. The vagina is protected by microbes that maintain an acidic pH. However, when these microbes are eradicated by antibiotics, the yeast Candida albicans can flourish. The urinary tracts of both males and females are protected by thick layers of epithelia, as we can see in the histology sample. Furthermore, urination flushes microbes from the urethra. However, the bacteria E. coli has special adhesion molecules that allow it to adhere to the urothelium. Attachment is especially likely if urination is obstructed. However, the bacteria E. coli has special adhesion molecules that allow it to adhere to the urothelium. This can especially occur when urination is obstructed. Next, let's illustrate an overview of how pathogens are disseminated through the body. First, indicate that pathogens enter through a breach in the epithelia or mucosa. Once inside the host, some pathogens infect peripheral nerves and travel along their axons to then reside within the neuron. For example, varicella zoster virus causes chickenpox, but then resides within the neuron itself. Upon reactivation, this can cause shingles. Other pathogens travel within macrophages and other cells or move freely in the plasma, show that dissemination via the lymph and blood vessels can lead to systemic infections or organ-based pathologies. Some pathogens, especially neurotropic viruses and certain parasites, are tissue trophic. This means they infect specific tissues and are therefore associated with characteristic disease patterns. Let's chart the course of a typical acute infectious disease in 5 stages. Create a graph to illustrate how the number of infectious agents changes over time. Indicate that the first stage is the incubation stage. This is the time between the acquisition of the pathogen and the onset of symptoms. Next, during the prodrome stage, nonspecific symptoms such as fever or tiredness occur. Show that later, as the host immune system and/or treatments effectively reduce the number of pathogens, symptoms decrease during the recovery stage, also called the convalescence stage. Ultimately, symptoms disappear and the individual returns to health. However, some may become chronic carriers of the infectious agents or develop latent infections that can reoccur. Finally, let's indicate some ways that infectious pathogens evade the host immune system, thus ensuring their own survival. First, write that antigenic variation allows microbes to change their surface antigens to evade antibody recognition. Recall that innate immunity relies on defensins and other antimicrobial peptides that insert pores into the microbes, causing their lysis. Indicate that some microbes, such as Staphylococcus aureus, resist pore insertion or eject the peptides. Phagocytosis, which is the uptake and subsequent killing of microbes, is effective against some pathogens. However, indicate that some microbes use their external capsules to avoid phagocytosis, disrupt phagolysosome fusion within neutrophils, or even directly kill phagocytes. Show that some microbes block the complement pathway and therefore prevent formation of the membrane attack complexes that provoke lysis. Viruses are targeted by cytokines, especially interferons, for removal. However, some viruses have evolved decoy receptors that weaken interferon effects. Another tactic is to inhibit pathways downstream of the interferon receptors. Furthermore, some viruses, including herpes simplex and Epstein-Barr virus, hide in latency, potentially reactivating when opportunities arise. Also, as we discuss elsewhere, HIV virions infect CD4 positive lymphocytes, which ultimately suppresses the immune system. Also, as we discuss elsewhere, HIV virions infect CD4 positive lymphocytes, which ultimately suppresses the immune system. Finally, denote that the host's own immune response to pathogens can be a significant source of damage in infectious disease. We learn more about immune responses and their potentially destructive effects elsewhere. This concludes our diagram.